Hi there, this is FemmeSciComm's 8th vlog, and today's post is going to be on fertility and the changes that happen with age. So it's not a surprise that more and more women are delaying childbearing and having children later in life. However, this coincides with a higher risk of miscarriage and having children with genetic defects. I myself I'm getting older and don't have any children yet, and it's a scary idea thinking once I'm ready to have kids, will I be able to have them? So today I'll be discussing a paper that assesses what age-associated changes happen in the ovary, specifically the fat tissues surrounding the ovary. This paper was recently published in Reproductive Biology and Endocrinology. Histologic analysis and lipid profiling reveal reproductive age-associated changes in peri-ovarian adipose tissue. The first author is Shweta S. Dipali, and the senior author is Francesca E. Duncan, both from the Feinberg School of Medicine at Northwestern University. So let's get into it. This study asks the question, how do the fat cells surrounding the ovary change with age? First, what do we know about fertility in advanced reproductive age? Or what happens to women's fertility as we get older? Research has shown that there's a downturn in not only the quality of our eggs, but also the quantity of our eggs. But who cares about the ovarian microenvironment anyway? The microenvironment is gaining more attention because it contributes to the maturation and health of the follicle. And as women get older, there are lots of changes that happen in the ovarian microenvironment. So the researchers of this paper hypothesized that age-associated changes in adipose tissues surrounding the ovary may impact ovarian function. First, where are the fat cells that surround the ovary? This research group uses a mouse model. Here is where the adipose tissue is that they aim to profile. The researchers found that the size of the fat cells, or adipocytes, were increased in mice that were reproductively old versus those of the reproductively young. Not only this, age was also associated with increased animal weight. This change in animal weight correlated with an increase in the amount of peri-ovarian fat that surrounded the ovary. They found that the difference in adipocyte size between the young and the old were statistically significant. The authors also looked at any differences in the lipid profiles of the peri-ovarian adipose tissue via MRM profiling. They specifically saw differences in the triacylglycerol or TAG lipid profiles. They determined that overall TAG profiles differ with age. The profiles of the tags themselves were different between the reproductively young and reproductively old in the length of the fatty acid chains, the saturation of the chains, and the composition. So what does this all mean? More and more women are delaying childbearing, which means that more women are having children at an older or advanced reproductive age. However, I've already mentioned that this coincides with a higher risk of miscarriage and or having children with genetic defects. But the mechanism and the other factors that contribute to this have not yet been specified. This study helps detail the changes that occur in the ovarian microenvironment with age. The researchers found that the fat surrounding the ovary undergo adipocyte hypertrophy in reproductively old mice. Not only that, but the composition of the fat itself undergo many changes in reproductively old mice. Specifically, the triacylglyceride or TAGs, also known as triglycerides. And triglycerides are the main constituents of body fat in humans. Aging and weight gain are known to associate with one another in humans. For example, with age, there's usually an increase in fat mass and visceral adipose deposition, the fat stored in the abdominal cavity near the organs. Also, did you know that obesity accelerates diseases common to old age? Obesity also affects fertility. This research group focuses on the physiological changes that occur in the ovarian microenvironment with age and in the future how that will affect fertility. But don't forget the caveats. Women do not have peri-ovarian adipose tissue, but women do have visceral omental fat, which is highly analogous to this. But it's unknown if it's functionally similar to the peri-ovarian adipose tissue that they profile. Another caveat is that this study has a relatively small sample size. But the authors have stated that this is a preliminary study and functional studies will follow to determine how these changes influence both adipocyte and ovarian function. Overall, this study profiles some of the age-associated changes that occur in the microenvironment of the ovary, specifically in the peri-ovarian adipose tissue. Changes in the ovarian microenvironment that occur with advanced reproductive age may lead to the age-related infertility that we observe around 35 to 37 years of age in women. And this study can help identify promising new targets for women in advanced reproductive age that want to stay fertile, especially in this era of delayed childbearing. Thank you for listening, watching, and reading my assessment on this paper on fertility in the ovarian microenvironment. Keep informed, stay interested, and keep doing great women's health research. If you're interested in this content, please make sure to subscribe to my page or share and like this post. I post every Thursday on FemmeSciComm.com, and you can also follow me on Facebook, Twitter, Instagram, and Pinterest at FemmeSciComm. Thank you. Bye.